Hello, everyone, and welcome to episode 635 of the People Behind the Science podcast. Today, we are excited to be sharing our conversation with Dr. Spencer Barrett, originally published on July 8th of 2015. Now, listeners, if you haven't had a chance to get to know Spencer yet, he is an evolutionary biologist and world-renowned authority on the ecology and genetics of plant reproduction. His work has investigated how flowers evolve and the mechanisms responsible for mating system transitions in flowering plants. And among his many accomplishments, Spencer's lab provided the first experimental evidence for the purging of deleterious genes after inbreeding in plants. And in our conversation, listeners, Spencer shares more about his career, his life, and his research. So get ready to enjoy this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am thrilled to be here today with Dr. Spencer Barrett. So, Spencer, welcome to our show today. How are you?
I'm very well, Mary.
Wonderful. Well, we are delighted to have you on our program today. And listeners, just a little bit about Spencer. He is the University Professor, Canada Research Chair, and Professor in the Department of Ecology and Evolutionary Biology at the University of Toronto. He completed his Bachelor of Science degree in Agricultural Botany from the University of Reading in England and received his PhD in Botany from the University of California, Berkeley, before joining the faculty where he is today now at the University of Toronto. And now Spencer has received many awards and honors during his career, including being named a Fellow of the Royal Society of Canada, a Fellow of the Royal Society of London, a Foreign Honorary Member of the American Academy of Arts and Sciences, and an Extraordinary Professor by the University of Stellenbosch in South Africa. He has also received the Lawson Medal from the Canadian Botanical Association, Premier's Discovery Award for Life Sciences and Medicine from the Ontario government, and the Sewall Wright Award from the American Society of Naturalists, among others. And he is also currently the Editor-in-Chief of the Proceedings of the Royal Society in Biological Sciences. Now, Spencer is here with us today to tell us about his journey through life and science. So, Spencer, today we're going to get to know you, both as a scientist, but also as a person. So can you start just by telling us what you like to do with your time when you're not doing science?
Well, I have to say probably what I like doing most is traveling. I mean, that sounds kind of a little bit corny because as an academic, of course, we always travel. But I've always enjoyed traveling ever since I was a teenager. I would say it's almost motivated some of my research in an effort to go to places that are exciting and exotic. So traveling is a big issue for me. I spend probably about 3 months a year away from the University of Toronto. And I've been fortunate enough to travel to many very interesting places, which I could tell you about later. So that's probably the thing I enjoy most. I've just come back from a month in Brazil, where I've been working in the Pantanal, as an example. But the other thing, and again, this is not perhaps too surprising, although I would say not all botanists like gardening, but I'm a very keen gardener. I don't know whether that's because I originally hail from the UK, where there's a great community of gardeners, but my father had a wonderful garden, so I grew up around flowers. I guess when I was younger, I actually rather resented being dragged around nurseries and things like that with my father when I was about 7 or 8, but it stuck, and I actually do love gardening, and my wife and I have a nice garden here in Toronto, and we potter around in it at weekends. We don't have a cottage, so we actually spend quite a lot of time just enjoying our perennial gardens.
Wonderful. So do you have a favorite flower or plant that you grow?
Well, that's an interesting question because I thought I did. I used to really, really love irises. Mm-hmm. I certainly developed a collection of irises in the 80s in this garden, but you sort of move on to other groups.
Gotcha.
Now I'm fascinated by tree peonies. I also love primulas, and I have ponds in my garden, so I'm really trying to develop a diverse set of aquatic plants, which I'm attracted to.
Very interesting. Well, I must say I'm a fan of irises myself. We've got quite the assortment in our own backyards. Wonderful to hear about your life outside of work. And I think you're right. Sometimes what you do at work is something that you also love doing at home. So you get to work with plants at home and at work, which is fantastic. But now I want you to tell us a little bit more about your work. So can you tell us how you describe what you do to someone who's not in science?
Sure. I think a lot depends on who that person is and how much they know about science. So, I mean, if it's somebody who has absolutely no scientific background, I usually just say I'm a biologist. Mm-hmm. But depending on that person, I would then go, say, evolutionary biology. I mean, that's basically what I am. I'm an evolutionary biologist. I specialize in what is called ecological and evolutionary genetics. But I'm also a botanist, and I've always loved flowers, plants. So I guess I'm a plant reproductive biologist because I study the ecology and evolution of plant reproduction. And one other thing I often tell people, because I think this is quite a legitimate thing to say, even in this day and age, is that I consider myself a bit of a plant explorer. There's nothing more appealing to me is to go off to somewhere exotic to find some interesting plant. So I love plant exploration.
Very interesting. I think that makes quite the romantic scene of your work, the plant exploration idea. And have you yourself found new species of plants when you're out discovering perhaps in Brazil or Peru?
Well, there's one species I found in Jamaica, which was then subsequently described. It was a turnera. I've certainly discovered some interesting issues to do with missing forms and things like that. And I was very fortunate when I was a graduate student to be in the Amazon, and I discovered a missing form of a very well-known plant, water hyacinth, which is one of the world's worst aquatic weeds. And Charles Darwin had sort of predicted that there should be this form, and I was lucky enough to find it. And it had a fairly big impact on my career because I then became obsessed with water hyacinth. And in fact, that's why I was down in the Pantanal working on water hyacinth. So it sort of stuck with me now for most of my career. That's just one example.
Well, that sounds wonderful. I must say I'm continually impressed by Charles Darwin's predictions. It's sort of, and the man thought of so many things, which is phenomenal. So great to hear a little bit about what you do on a daily basis as a scientist. And now I'm curious to take a step back and talk about motivation. I think there's so much to do in a day that sometimes it's nice to have these little motivational quotes or sayings to keep you going. So can you share with us today one of your favorites?
One that I really liked, which I think certainly influenced me in making a decision to work on plants as systems to study evolution and ecology, was one by John Harper. And John Harper was a very famous plant population biologist who wrote an influential book in 1977. And one of the things he said about plants, and I mean, it's a very simple quote. He just said, "Plants stand still and wait to be counted." And why that's important is that often it's very hard to collect data from various groups of organisms. Mm-hmm. But that's not necessarily the case with plants. They're immobile and they just sit there and they're ready organisms to study and collect data without too much pain. I once ran into a group of scientists in the Amazon who were studying the manatee. And when I asked them what was their sample sizes like for the kind of work, they'd only seen 2 or 3.
Oh, wow.
Whereas with plants, you have an abundance of data, and that makes life a little more easier. So I rather like the idea of plants standing still and waiting to be counted.
Well, I love that quote. And I must say, the one field project I have done in my career, I did choose plants instead of animals for the exact same reason. So.
Well, I should say on field courses, I teach a lot in the tropics. And when we have a group of students from University of Toronto in various locations, generally speaking, most of the students are not very interested in plants. They want to see monkeys or exotic animals in the tropical rainforest. Mm. But when it comes down to then actually doing projects, because we require the students to do projects on these field courses, reality sets in, and it's remarkable how many of them end up working on plants.
Right. When you have to think about the logistics of it, plants seem like a very, very good option. Well, Spencer, great to hear about one of these little quotes, tidbits that has had an impact on you in your career. But now I want to talk about some of the people that have motivated you and inspired you over the course of your journey. So can you share with us today who you consider your role models?
Yes, I thought about this for some time in terms of another context that somebody asked me about role models. And I would have to say, at a grand scale, Charles Darwin never ceases to amaze me. Mm-hmm. He was the founder of plant reproductive biology. It's not widely known that Darwin spent more of his life working on plants than he did on animals. Pretty much after he came back from the Beagle and started living at Down House, he became fascinated with plants, and throughout the time he lived, he did experiments. He had a glass house, and he wrote 6 books on plants, of which 3 were on plant reproductive biology. And these books are not books that are dusty in the back of some cupboard in my house.
Sure.
I frequently refer to them because they still have an amazing set of insights which are relevant today. I mean, his ideas have been remarkably durable with regards to many of his ideas about plant reproductive biology and the benefits of outcrossing and selfing and so on. So I'd have to say at the big scale, it would be Charles Darwin, but then ratcheting forward, I was very influenced by a wonderful high school teacher that I had, Muriel Hosking, who's still alive in England, where I grew up. And she was the model high school teacher in the sense that she was absolutely committed to teaching us biology and particularly taking us into the field, which I guess is probably harder to do now with all sorts of security issues. But almost every weekend I would go out with my school chums with Muriel to the chalk downland or badger watching, and that really got me excited in natural history. So she was a very important early influence on my life.
Well, that sounds like a great opportunity to get out there and start doing field science in high school, which I think there are not many people out there, you're right, who have had that opportunity, particularly in the later years. So very interesting to hear about your experiences. And I love this description that you had about Darwin and still pulling out his work today to get inspiration, to get ideas. And I think it's interesting now that we have so much more advanced technology to have the opportunity to start looking at Darwin's ideas more precisely. So is this something that you seek Darwin's inspiration and then turn this into projects in your lab?
Oh, absolutely. I would say the majority of projects that I work on, the fundamental question was probably promoted, or at least the idea came from work that Darwin did during the time he was working on plants. Much of my research career, I've focused on a particular plant sexual system known as heterostyle, and Darwin devoted 2/3 of one of his books to heterostyle, and he was very interested in really asking questions about what was its function, how did it evolve, why does it often break down to selfing. And when I read Darwin and realized that this would be a wonderful experimental system to apply modern techniques to, I got sort of really attracted to studying heterostylous plants and still do today.
Certainly. Well, very interesting to hear about this example. And I'm curious now to hear about where your interest in science began. I mean, you mentioned this phenomenal high school teacher who was able to really bring you out in the world and do hands-on science with you. But did your interest extend further before that?
Yes, I would have to say that my family, growing up in England, my mother and father, they used to love gardening, and that initially exposed me to being around plants and a diversity of different kinds of plants. And as I mentioned before, we would often spend the weekend going to a nursery or a botanic garden. And then the other thing, of course, is my parents, they really enjoyed holidays, and particularly camping holidays. So growing up in the UK, we used to initially go to the south of England, and then my father took the big and bold step that we would go to the continent,
Ooh.
which was very exotic in the early 60s. And we traveled around Europe quite a bit. And I would always take the opportunity to go off and on a natural history walk. So I think my early interest, I mean, I was a bona fide natural history geek as a kid. Mm-hmm. I used to love butterflies, I loved plants, I loved beetles. There was this thing called hobbies in those days.
Right.
Maybe are not so common now, but I was into all sorts of hobbies when I was a kid that involved different groups of organisms. So that was the beginning. And then I went to the University of Reading to do my bachelor's degree. And the teachers there were very good, and I had some wonderful opportunities. Probably the most pivotal moment was when the professor who was teaching me, who was the professor of the department, A.H. Bunting, raised the issue that would any of us like to take a year off from our undergraduate degree and go somewhere for a year. It was termed an intercalated year.
Oh, wow.
And sort of do research. And so between my second and third year, Bunting was able to place me at a research station in Swaziland in Africa, which you can imagine, as I was 21 or 22 years old, this was an incredible opportunity. And so I left home and went to Africa for a year. And I think that was really the turning point in my life when I realized I want to do research. I don't know enough. A bachelor's degree isn't going to be enough. I better seriously think about graduate school. And that year in Africa had an enormous impact on me for a variety of reasons. First of all, I was exposed to amazing tropical diversity and saw all of these plant groups that I'd never seen before. Secondly, made me interested in politics because this was 1969, 1970, and I spent a month hitchhiking around South Africa because I wanted to see the landscapes and the biology, and of course the apartheid regime was in power then.
Right.
That had a big effect on me. And then when I finished in Swaziland, I spent 3 months traveling back through Africa. And I was very fortunate to go to many countries, Malawi, Kenya, Tanzania, Ethiopia. And again, it just inspired me that there was a lot out there that was very different from London and the UK, where I was sort of brought up. And probably more than anything else, when I was in Swaziland, my chums there, my best friends, turned out to be Peace Corps volunteers who were working in Swaziland on various community projects. And I have to say, I really enjoyed interacting with them. I didn't have a very high opinion of Americans at that stage because of the Vietnam War. I protested outside the embassy in London and wasn't very happy about what was happening in that particular war. And then I met all these really interesting Peace Corps volunteers and came to the conclusion that actually living in the States would probably be okay.
Right. We're not half bad.
So anyway, I basically decided to explore the possibility of going to graduate school there. And then I spent 3 months hitchhiking around the States one summer, visiting the various graduate schools where I'd applied and got accepted. And I have to say, when I got to the far west, that was it. I was smitten by California, and I decided to do my PhD there, and I've never regretted it. It was an absolutely wonderful place to do a PhD. And then other opportunities there. My supervisor, Herbert Baker, was a wonderful person, just the nicest man. And through discussions of various projects, I got the opportunity to go to the Amazon to do my PhD. And that's where I had this fortuitous meeting with the short-styled form of water hyacinth. I mean, basically, I went down to the Amazon to look at the origins of weed floras, because in the Amazon, they were starting to put in rice. And there was this opportunity to see, well, what kinds of plants colonize these rice fields since they were starting from scratch. It's often very difficult to study, if you like, the origins of a wheat flora because agriculture has been around for some time.
Right.
Certainly,
they're everywhere.
It's hard to do it in Europe, where it's been around for 10,000 years. So my initial idea was to go down and sort of try and understand the origin of a weed flora, but then by chance, I bumped into this water hyacinth, and I actually didn't even know the species because I'd not been familiar with this group of plants because they didn't occur in the UK. But I had been very lucky when I was at Berkeley to have taken a course from a professor who had discussed this controversy with Charles Darwin and how he'd been sent material by Müller, the famous German naturalist, or one of them. There were two, Fritz and Hermann, and they were down in southern Brazil, and Darwin was a great correspondent. And the Müllers had sent this material of water hyacinth, and from these herbarium specimens, Darwin inferred, or he deduced, that this species must be what is called tristilus, which is where there are three sexual morphs that coexist within a population. And the only way outbreeding can occur, normally with this system, is crosses between the forms. So Darwin made this sort of suggestion, but subsequent to that, nobody had ever found the short-styled form. And it was a sort of a bit of a controversy, and there was some suggestion that it might have gone extinct. And then, lo and behold, the first plant of water hyacinth that I picked up in the Amazon actually had this short-styled form. In other words, the female sex organ is short in length, whereas the female sex organ, which is known as the style or the pistil, on the other two forms are medium length and long length. So, yeah, it was a pretty good start.
So what was going through your mind when you picked up this almost long-lost sounding flower form?
It was a bit delayed because I was uncertain as to whether, in fact, this was Eichhornia crassipes, which is, in fact, the species that Darwin talked about, because there are other Eichhornias in the Amazon, and some of them look quite like water hyacinth. They are distinct species, but I wasn't quite sure because I'd never seen this plant before. I had just got married. My wife, Suzanne, was actually with me, and she was doing her graduate work in the Amazon as well on planning of national parks in Amazonia. She's an environmental planner, and she was traveling around the Amazon to various places looking for potential sites for national parks. And so she left me at this research station and set off with these herbarium specimens. And I said, when you're in Belém, go to the herbarium and see if you can get this identified. And she didn't come back for a month.
Oh, wow.
We had no communication with one another. This was before email and...
Right. Cell phones, yeah.
So I was quite excited when she did return, not only to see her, obviously, but that she told me, oh, yes, this is actually Icornia crassipes. So that was when I celebrated. Then I had the issue of informing my major professor that the riceweed work, should it take a back seat? I've just discovered Darwin's missing form. And Herbert was wonderful. So I wrote him a handwritten letter from the Amazon. This was in 1973, I think it was, or 74. It took about 3 or 4 weeks before I got this letter back from Herbert with the news that drop the rice weeds and focus on Darwin's missing form.
That is a phenomenal story, Spencer. I love this description of a almost serendipitous encounter with this water hyacinth that really ended up shifting your research trajectory. So very, very cool. And I think when you see those opportunities as a scientist, you have to jump on them. You can't just say, well, this is interesting, but this rice project, you know.
I mean, I haven't given up on water hyacinth. The whole reason for going to the Pantanal in April, I spent April wandering around in chest waders in the Pantanal wetlands, actually sampling water hyacinth clones. So I'm still working on water hyacinth.
They keep coming back. Yeah. I like it. Well, wonderful to hear about your journey, Spencer. But now I'm curious to hear a little bit more about what you're working on today. So you mentioned the water hyacinths are still in the picture, but can you describe one of these projects that you are doing now that you are the most excited about?
Well, I think if I had to define for you, the core research in my lab is about evolutionary transitions. So that's about finding variation in nature where in some populations the plants are reproducing in one way, but in other populations of the same species, they've actually evolved a different strategy.
Interesting.
And the one that I'm particularly interested in is the evolution of self-fertilization from cross-fertilization. So most plants are outbreeding. Outbreeding generally maintains genetic variation in populations, and the majority of flowering plants are outbreeders. And we actually know, going back to Darwin, that the evolution of self-fertilization is to some extent a sort of surprising thing, because Darwin was able to show, again, another book that he wrote, that the products of self-fertilization generally are a lot inferior to the products of outcrossing. So progeny that come from outbreeding are usually more vigorous, perform better than the progeny that we obtain if we artificially self-fertilize a plant. And we know the reason for that now. It's because of what is called inbreeding depression, and it's simply that those self-offspring are generally homozygous for deleterious genes, which in the outbreeders are sheltered in the heterozygous conditions. So we sort of know quite a bit about the genetic architecture of inbreeding depression. But the question then becomes, well, do we find species that naturally self in the wild? And we do. They're not as common as outcrossing species, but there are probably around 10 or 15% of flowering plants self-fertilize predominantly. So a critical question has been, how does this occur and why? And so I've been working on this problem for some time, but what has happened recently, which is very exciting, is that we've been able to sort of apply genomic tools to this question. As you know, biology is being revolutionized by high-throughput genome sequencing and the opportunities for looking at very large numbers of genes now, whereas before...
It was a long process, yeah.
Yes, and I often say that when I started my PhD, I was working on 1 or 2 genes. These were the genes that were controlling this particular sexual system, so that was well-defined genetically. I then, in the 80s, increased the number of genes I was looking at because protein electrophoresis was very popular, and it now became possible to screen 20 or 30 or 40 genes. Now we're looking at thousands and thousands of genes. And
you're almost a kid in a candy store, yeah.
It's been a very steep learning curve for an old geezer like me. Because now I generally sort of look over the shoulder of a postdoc or a PhD student who is staring at the computer screen, working on a pipeline, trying to isolate SNPs from genomic data. And generally all I can do is give encouragement.
Right.
So
Pats on the back.
But in any case, I'm fortunate to have some really great graduate students who are working on a set of problems. And the particular problem that we've just published on, which I'm quite excited about, is that my student Ramesh Arankumar, who's doing a PhD with me, has been studying this transition from outcrossing to selfing in a relative of water hyacinth. This is an annual species that we've used as a model system now for about 20 years. And by doing genomics on outcrossing and selfing populations, that is, looking at the whole genome now and asking what are the genomic consequences of this transition from outcrossing to selfing, we've been able to make some quite interesting discoveries which are quite exciting. For example, we have evidence in the selfing populations, in the genomic data, of what is known as genetic purging. This is the idea that if a plant selfs for some time, then deleterious genes that do arise, which are now going to be in the homozygous condition, can be purged from the genome by what is known as purifying selection. And so we now have evidence that this can occur right across the genome in these selfing populations. And that's quite exciting because in the early 90s, Deborah Charlesworth, a colleague of mine, and I published a paper in Nature where we were able to demonstrate purging of the genetic load for phenotypic traits, so observable morphological characteristics of the plants that were related to fitness. But now we've been able to sort of extend that to evidence for purging right across the genome. So that was quite exciting to get that evidence.
Very interesting. So with this idea of purging being present, do you think there will be more plants potentially switching over more than that 10 or 15%? Or why don't we see more than 10 or 15% if they're able to get rid of those deleterious genes?
It is possible to have purging, but the early stages of that transition, because of inbreeding depression, often many selfing lineages will evolve, but then they go extinct.
Gotcha.
There is fairly good evidence that the transition to selfing can be quite ephemeral, and that selfing lineages, we talk about selfing as an evolutionary dead end. In other words, it may provide short-term benefits, and those benefits I haven't talked about, but the most obvious one is that if a selfing variant arises in an outcrossing population where pollinators are unreliable, or let's say a selfing variant is dispersed to a region where there aren't pollinators that visit this particular species, and that can happen, for example, by long-distance dispersal to islands. That's, by the way, exactly what happened in this Icornia. All our populations, or the vast majority of our populations in Brazil, are pollinated by specialist long-tongued bees, which are abundant there. But there has been a transport of seeds, probably by waders, and we dated it at around 125,000 years ago. There was long-distance dispersal to the Caribbean, and there in the Caribbean, all of the populations are highly selfing, and they're not visited by long-tongued bees, the specialist long-tongued bees, because there aren't any there that visit these populations. So selfing does have these short-term benefits. And in a lot of cases, they'll pop up on the landscape, hang around for a while, and then because of inbreeding depression and other problems to do with the effects of selfing on the genome, those populations go extinct.
Well, this is a fascinating area of research, Spencer, I must say. And we've chatted a little bit about the adventure that you've had going through science and kind of touched on one of these challenges in terms of the changing technology in science and how to cope with it. But I know every scientist has dark days, challenges, failures, things like that. And we don't always hear about them, and the public doesn't always hear about them. So can you share with us today, Spencer, one of these challenges or failures that you've experienced and how you got through it to be successful?
I've been pretty lucky. I mean, I haven't had any really devastating failures. I've heard of people's having data stolen or labs burnt down and things like that. I mean, something that is really
catastrophic.
Yeah. But I would say that as somebody who does a lot of work in the field, both experimental work and survey work, often things don't go quite as planned.
You don't always reach in and grab that long-lost flower every time.
Yes, I have two stories about that. I mean, one is that I was very interested in going to Panama to find a particular species that was going to be very important to a story I was trying to develop in this group of water plants that I work on, the water hyacinth family. And I planned this trip. I thought I knew exactly when this plant was flowering, and I went all the way down to Panama. and the thing wasn't in flower. So if it's not in flower, we can't do the sort of work we wanted to do.
Sure.
So that was disappointing. But probably even another version of that was in 2003, I took a sabbatical in South Africa, which is one of my favorite places to do research in the Cape. And I had a very specific project that I wanted to work on there involving looking at the function of what are called mirror image flowers. These are flowers that the population produces two kinds of flowers. One set of flowers has the sex organs deflected off to the left and the other to the right. So it's a wonderful example of asymmetry in flower architecture. And one of my PhD students, Leslie Jessen, had done a wonderful thesis on these plants in South Africa at the end of the 90s, and we had done field work there together, and we had all these other questions that we wanted to address. And in the meantime, Lindley had left and got a job in New Zealand, and I wanted to pursue this work. And I went down to the Cape at the time that these plants reliably flower, and they weren't flowering.
Oh, no.
And this wasn't a short-term visit, right? This was a 6-month sabbatical.
It's not just a quick stop away. It's quite a distance, too.
Yeah, I had anticipated working for a couple of months on this. So that was really disappointing. But on the other hand, often these things, there's a silver lining, because what actually happened was that I was then looking for a project. And I met up with a graduate student at the University of Cape Town, Bruce Anderson, who's now a faculty member at the University of Stellenbosch. And Bruce was a great natural historian, loved to go into the field. And I said, would you mind if I hung around with you and that we could go out together and you could teach me some of the plants I don't know? And on one of those trips in the Cape, we stumbled on a really interesting plant, and then ultimately were able to solve a really interesting problem. So the story is about this plant has a bird perch that it's evolved. It has a structure which is a very strange structure. It's a cylindrical, looks like a stem that comes from the bottom of the inflorescence. The inflorescence is simply a cluster of flowers. And the flowers are actually on the ground. So this plant has ground flowering, which is very unusual. So the flowers are displayed on the ground. They're very showy, bright red flowers. And we were wandering through the fynbos, and I said to Bruce, what is that? That's a very strange plant. And by the way, it's in the iris family. It doesn't look anything like an iris, but in fact is in the Iridaceae. And the genus is Babiana, and actually that genus is quite common in the Cape. And so we looked at this thing, and I said, well, what is this structure? And he said, well, funnily enough, the plant's common name in Afrikaans, translated, is rat's tail. It looks like the tail of a rat, this dark structure that simply comes up vertically from the flowers, which are at ground level. And so we looked at this thing and said, there's got to be a problem here to work on. And I said, well, the flowers, they're on the ground. What do you think pollinates these? They look as if they should be bird pollinated. What pollinates this thing? A chicken? I mean, if we're North American, we're of course familiar with hummingbirds being major pollinators of flowers.
Sure.
They hover while they pollinate the flowers. And most of the flowers are produced above ground and accessible to the hummingbirds, which then visit them for nectar. But of course, there's no hummingbirds in the old world. And all of the birds that pollinate there, primarily sunbirds and honeyeaters, instead of hovering, they perch. And so we slowly looked at this thing and said, this plant has evolved a perch.
That is bizarre.
Which allows the potential pollinators to visit the plant and access the flower. And so anyway, what we ended up doing was sort of some manipulative experiments in the field. We used genetic markers. We looked at what was the adaptive significance of this structure. And we were able to show that, in fact, this plant has the most specialized bird perch in the world. And there's only one pollinator that visits it, the malachite sunbird, which is a rather large sunbird. And these animals just simply flew in, grab hold of the perch, turn upside down, and then probe the flower for nectar. So we didn't find any mirror image flowers, and that project didn't go anywhere. But we found another one, and that's the wonderful aspect of field biology. If things don't work out, go and find another project.
Right. Keep looking around. There's something around you, I'm sure, that is an interesting story behind it.
Right.
So, Spencer, I love this story of how you turned a failure, a challenge, into a success. And I think this sort of ingenuity, this flexibility is an important trait in scientists to be successful. Like you said, you're not going to be lucky every single time. You're going to run into these failures, and you need to be able to react in a way that you're able to turn it into something that's not a complete failure. So I love that story. And we chatted a little bit about your work throughout this interview, and I'm curious to talk now about some of your life outside of science. So we mentioned that you enjoy getting outside and gardening, but I've met so many scientists who are quite avid readers. So Spencer, I want to hear what's on your bookshelf. Do you have a recommendation for myself and our listeners in terms of a good book?
I'm going to recommend two. I mean, I should say I am an avid reader, particularly when I'm traveling. I mean, that's how I get over long airline flights to China and to various parts of the world that I visit.
Sure.
And usually when I'm flying on those flights, I like sort of P.D. James and Ian Rankin and Ian McEwan, a good paperback, which I can get my teeth into. But I want to mention a book that was a real page-turner that I read several months ago, which I could recommend to the listeners. I couldn't put this book down. And I have to say, the book is related to evolutionary biology, but it's a biography of W.D. Hamilton, written by Ulrike Segerstrahl, and it's called Nature's Oracle, The Life and Work of W.D. Hamilton. It's one of the most wonderful books I've ever read because it takes you behind the scenes and almost you get to feel you really know the character the book is being written about. W.D. Hamilton is considered by some to be the greatest evolutionary biologist since Darwin. He was an amazing person. He was at the University of Oxford. He's most well known for his ideas and his theories on kin selection. But he was also an amazing naturalist, and he used to go off to Brazil, particularly, very regularly for inspiration. But why the book is so interesting is that he had a rather troubled relationship with another great evolutionary biologist, John Maynard Smith. And this book sort of takes you behind the scenes on this relationship. Both of these individuals have passed away, unfortunately, in the last decade, both tragically. And all of the correspondence and all the full story of their interaction is laid out in exquisite detail in this book. I wasn't personal friends with either of these people. I met both of them and interacted quite a bit with John Maynard Smith to actually read about why their relationship was troubled and how this affected so many aspects of their persona, particularly towards their later years, was fascinating. And I would recommend that for a really great biography of a very important mind in evolutionary biology. On a totally different note, I've also just read, because I like politics and I'm particularly interested in South African politics, I've just read a book about Helen Suzman. It's called Bright Star in a Dark Chamber by Robin Renwick. And Helen Suzman is a hero of mine because she was one of the very few people who stood up against the apartheid government in the parliaments in South Africa. She was a close personal friend of Nelson Mandela, and this book charts her life as being one of the very few elected people who was able to stand up in parliament and stand up against the leaders of the Nationalist Party. It's an excellent book, and it gives you great insight into a wonderful woman. She has passed away also, and she passed away in 2009.
Well, fantastic recommendations, Spencer. I will put those out on our website for our listeners to find. And I really like how you're emphasizing the stories behind the people, the stories behind the science, and particularly interesting that you actually knew some of these scientists, and then maybe it all made sense when you read the rest of the biography, kind of the intricacies of the relationships there. So very cool. And we've talked throughout our interview about some of these opportunities you've had to travel for science. And I'm going to ask you a tough question, Spencer. I want you to tell us about your favorite place that you've been for science. And what was it about this place that made it your favorite?
There's so many incredible places. I mean, I'm not going to give you just one. I will tell you where my favorite place is. New Caledonia had an enormous impact on me when I visited there. I wanted to go there to see the very basal angiosperm species, the sort of most, quotes, primitive angiosperm species, and also to see all the endemic gymnosperms that are there. It has the highest level of endemism of gymnosperms of anywhere in the world, and it's this tiny island out in the Pacific. Absolutely stunning place. I've also had the opportunity to go to the Tibetan Plateau and look at some of the alpine plants there. And China is an incredible place with amazing biodiversity. I love wandering through the Swiss Alps in summertime. I like to try and go there. I'm going to go there for several weeks this summer because there's nothing like being in a meadow, an alpine meadow in the Swiss Alps. But if I had to identify one place as my favorite place to go and to be, it actually would be the Cape, and particularly the drive from Cape Town up to Namaqualand at the time of year, which is August and September, when all of the flowers there are out. In other words, the season for seeing floral displays is August and September.
Interesting.
And particularly in a good year, it is absolutely stunning. There's nowhere else like it in the world for just sheer diversity of flowers. And I don't think I'm ever as happy as I am when I'm just sitting down cross-legged in Namaqualand, just looking at this amazing floral diversity, which appears, many of these things are quite ephemeral. Sometimes they don't come up, but if you have good rains, there's nowhere like it in the world to go. So I like South Africa for many reasons. I like the culture there. I like the wine. I like the beautiful topography of Cape Town and the amazing diversity. But that particular drive from Cape Town up to Springbok is my favorite thing that I like doing.
Well, I think you painted a phenomenal picture of just what it's like to be going on this drive surrounded by flowers. It sounds absolutely stunning. So great choice there. And thanks for sharing some of these travel opportunities. I think it is one of the fantastic things about careers in science is being able to see the world, being able to share your science and meet people from all over the world. And I think as a scientist, you do get an opportunity to work with some great people. And there are stereotypes out there about what scientists are like, but we always try and break those on our program by looking at the human side of science, the lighter side. And I think in labs, in these groups that you work with over the course of your career, there's a lot of funny memories that you can look back on fondly or just quirky traditions that spring up while you're there. So can you share one of these with us today, Spencer? One of these memories or traditions that you have?
Well, one of the traditions we have in the lab is that because I'm lucky enough to have a rather nice garden, we like to have garden parties. And so every year, at least if I'm here in the summer, I like to have a party for my lab and then other close friends. And one of the sort of things I like to do is play the hat game.
What is this hat game?
So what is the hat game? Well, for the last 20 or 30 years on my travels, I've been collecting hats. I like hats.
Any type of hat?
Oh, just whatever looks interesting. I mean, anything from, I have a New Zealand policeman's hat to hats from Brazil, hats from China, hats from everywhere. Wherever I'm at a place and I see an unusual hat, usually an authentic hat that is being used by people either in a workplace setting or maybe a ceremonial hat, I like to buy it. And of course, when you start to build up a collection of hats, people are very generous, and then I have friends who bought me hats and donated them to the collection. So I have a room in the basement where I have my hats. And so one of the sort of funny things we do with the hats is that when we have people over, I like to put a hat on somebody or everybody's hat because it has a rather dramatic effect on the personality of the individual. Actually, many of my graduate students have got wise to this now, so they actually bring their own hats and therefore don't have to suffer the hat that I give them. So that's one sort of funny thing. And actually, sometimes what we do is we even play a game at the dinner table. This is with a smaller group where people are blindfolded or they close their eyes, and then I will give them a hat. And then the game is that you have to guess the hat by asking questions that are, is this a hat that comes from Asia? And you're allowed 3 negative answers. If you get 3 answers wrong, you're out of the game.
Wow, this is very elaborate. I like it.
If you want to see some of the pictures of my students and colleagues with hats on, if you go to my website and look at Party Time, I think you'll see some of the hats there, and you can see some of them are pretty bizarre.
Listeners, we are going to have to check out these hats. It is official. So how many would you say you have, Spencer?
Probably, well, over 100. I would say probably about 120.
Wow. So you really do need that room dedicated to the hats, yeah?
Yes. Some are small. You could put them in a drawer.
You can stack them, yes.
You could probably imagine I have the classic hat that you see from tourists coming back from Mexico.
The giant sombrero. And
I've got one of those and it doesn't fit in the drawer very easily.
Well, that is phenomenal. I love these traditions that you've described, and I really like chatting with scientists about the different personalities of the lab and just these fun traditions and fun things that you do with each other outside of the lab to kind of hang out and get to know each other. So this sounds fantastic. And we've talked a lot about your work throughout our interview, and I want to know, Spencer, about these questions that you have not yet had a chance to tackle. So can you share with us today, Spencer, if we gave you everything, if things like funding and staff and technology and feasibility were not a problem, what is the question that you would want to answer?
One of the projects we're working on right now and putting quite a bit of effort into is about sex ratios in plants. And so there are a relatively small proportion of flowering plants that are not hermaphrodites, that have separate sexes. So there are males and females in populations. And generally one expects, based on theoretical predictions, based on negative frequency-dependent selection, that the two sexes should be at equal frequencies in populations. But we did a study a few years ago, this is a couple of postdocs of mine, David Field and Melinda Pickup. They carefully reviewed all of the literature to see whether, in fact, plants fit the one-to-one expectation. And it turns out that there are some plants where you do find one-to-one sex ratios, but a lot have biased sex ratios. And we were particularly interested in the causes of these biased sex ratios. And of course, they can be either male biased or female biased. It turns out the male bias is a relatively trivial answer. It's simply that males flower more often than females, or they're less prone to gender-based mortality. Females have the cost of making fruit and seeds, and that makes them quite prone to pathogens and diseases. So they'll often die more often than males, or, as I said, males will tend to flower more frequently, again, because their reproductive expenditure is less. But there are some plants that have female biased sex ratios, and that raises the question, what is the mechanism responsible for female biased sex ratios? And a lot of the work in the lab right now is focusing on that, and this has taken us into a direction that I never predicted we would have been working on, say, 10 years ago. But we're now looking at basically sex chromosome evolution, looking at plants that have sex chromosomes. It turns out sex chromosomes are not particularly common in flowering plants. Only about 12 to 13 families have sex chromosomes. But in this comparative analysis that David Field and Melinda Pickup did, there was an interesting association between female biased sex ratios and sex chromosomes. And so we've sort of been mining in on this and looking at a concept which was in the literature for many, many years and was not really taken very seriously, and that is the concept of surtation. It's basically a term that really means competition between pollen tubes in the style for fertilization of ovules. And so way back, a German geneticist called Correns found that depending on how much pollen you put on the female receptive stigma, he could alter the sex ratio of offspring. And the more pollen he put on, the more female biased the offspring were. This is a very interesting result.
Yeah.
And the question is, what is the mechanism?
Sure.
And the idea that Correns and then another scientist, David Lloyd, suggested was that, well, it may be that female pollen tubes, they're not female in the sense that gametes don't have a sex, but we can think of them as female determining pollen tubes. In other words, pollen tubes that are carrying the X chromosome versus pollen tubes that are carrying the Y chromosome. They're the male determining ones. It could be that the male determining pollen tubes are less competitive than female. So this is kind of Correns' idea that there was simply competition going on between these alternative pollen tubes, and females were more successful in fertilizing ovules, and hence you had more female progeny. Anyway, that idea sort of went into disrepute for one of the systems because it was reinvestigated in the white campion, Silene, and shown that actually another mechanism can probably explain this. But the other plant group that Correns worked on was sorrel, the genus Rumex, and we've actually now repeated his work, and we can actually show this effect. In other words, there is evidence that depending on how much pollen you put on the stigma, you're going to influence the sex ratio, and more pollen means more female bias. So the question then is, what is going on here and what is the mechanism? One possibility is that there are deleterious genes accumulating on the Y chromosome, And then when the plant makes haploid gametes, they affect the function of pollen tube growth. So that's a hypothesis. We do know now, through the work of a graduate student in my lab, Josh Huff, that there is evidence in rumex of Y chromosome degeneration. So that's quite commonly found in the Y chromosome. But what we're struggling with is how we link that Y chromosome degeneration to the difference in competitive ability of male and female determining pollen tubes. And we don't know how to do that.
Small detail there, yeah.
But I think it would be quite costly because it would involve looking at essentially distinguishing between pollen tubes in the style and determining which ones are male and which ones are female. I mean, we've made some progress on this. We now have large numbers of sex-linked genes, but how we would actually technically go into the style and figure out, oh, look, here's a set of fast-growing pollen tubes and they're female, and here's some rather slow-growing pollen tubes and they're male, that's quite a challenging technical problem. So maybe if I had tons and tons of grant money and an inordinate amount of time, we could solve that problem.
Sure. Well, this is quite an intriguing evolutionary puzzle that you've laid out. And I like how you sort of have some potential hypotheses and ways that you might be able to go about answering it. So very cool to hear about one of these big questions that's still lingering in your mind, Spencer. And we've talked about your work. We've talked about your path to get to where you are today. And I think over the course of your career as a scientist, you get to work with some phenomenal people, as we said. And some of these colleagues, some of these mentors, some of these even students that you have are great sources of advice. So can you share with us today what you would consider the best piece of advice you have ever received?
Again, hard to nail it down to just one piece of advice. I would say as an undergraduate, one of the faculty at University of Reading, Roy Snaden, he's long retired, was a very influential person for me because his attitude was that even though we were undergraduates and obviously we were reading lots of papers, he engendered a critical approach to the literature. In other words, don't believe everything you read in scientific papers. Mm-hmm. And I think that's been very helpful. I think that often scientists that have not come through strong programs, perhaps there may even be some tendency in certain countries where people tend to believe everything that's in the literature is true. And I think Snaden was great in making us look very critical. We had these reading groups where he would essentially take a paper apart and show that, well, this was okay, but this is a bit suspect.
Right.
So he was very important, I think, for a start. I would have to say my colleague David Lloyd, who was a New Zealander and really the founder of modern plant reproductive biology. I mean, Charles Darwin was the legitimate founder of the area of plant reproductive biology. But in the modern era, Lloyd was a towering figure who brought theory to the area, but he was also a great natural historian, so he was doing empirical work as well as theoretical work. He was a great mentor to me and gave me one piece of advice fairly early on in my career, which I think it's sort of affected the way I've approached graduate student training, and that is that to be successful in ecology and evolutionary biology, it's worth trying to pick a problem of general significance. There's so much organismal diversity out there, one can get sort of completely swept away in the detail.
Right.
But it's important to try and link one's research to general questions, particularly those where there is, in fact, some theoretical advancement, so that there's theory that can be tested. And I tried to do that in my work, and Lloyd did this very successfully. I guess the other thing he pointed out to me, that if you want to be successful, it's worth really burrowing in on one problem for a reasonable amount of time. He said a decade. Put a decade into, for me, it was heterostyly. I mean, he saw that as a really interesting general problem, and he encouraged me to work intensively on heterostyly for at least 10 years. Well, actually, I'm still working on it. So,
You're going above and beyond, Spencer.
Yeah, that's not to say one doesn't get interested in other projects along the way, but to have this core research interest, which can link to theory, I think was very good advice.
Well, wonderful advice to pay forward to our listeners out there who might be going through their scientific careers in search of advice. And Spencer, is there any other last piece of advice or any note of inspiration that you would like to end our interview on today?
Yes, I would. I mean, there's something that I think that is characteristic of the field of evolutionary biology today, and that is that we are being swept up in the genomics revolution, and it's extraordinarily exciting. I'm excited by it, and I think now we have the opportunity to address a whole series of questions that were impossible 10 or 15, 20 years ago. Of course, genetics hadn't been rediscovered when Darwin was around, but I just wonder, Darwin would have been absolutely mind-boggled, if there's such a word, by what we can do now with genomics. So I'm very excited about that. But I have a warning about it. It's my firm belief that we shouldn't forget the organisms involved, and we shouldn't forget good old-fashioned natural history. Because many of the really interesting questions in evolutionary biology come from observing organisms in the wild. And I'm a little worried that today's students are so focused on pipelines, genomic data, and just getting swept away with this mass of information that we're getting, that they're sort of giving up a little bit on organismal diversity. I don't think we're doing as good a job as we should in making sure that students take field courses, take systematics courses, so that at least they can tell the difference between a bryophyte and a fern. Mm-hmm. I mean, I do worry that many of our students, even in our program at Toronto in evolutionary genetics, they're a little bit nervous about their ability to identify organisms. And I think it's important that we make sure that they appreciate that natural history, old-fashioned Darwinian natural history, is at the core of everything we do. And so that by all means, become specialists in genomics, but go for a walk now and again.
Right.
Go and look at things.
Get away from the microscope, away from the arrays.
Yeah, don't spend your life staring at a computer screen trying to analyze DNA data. It's great that that is being a major focus, but it shouldn't be entirely at the expense of organismal biology.
Well, fantastic advice, Spencer. I know it is hard not to get that tunnel vision, but you do need to put everything in context of the real world and keep that in mind. So wonderful point to leave our listeners with. Can you tell them now how they can reach out if they want to learn more about you and your work?
They can visit my website. If you just put my name into Google and go particularly to my lab page, there's a lot there, and they can find out all about the sort of research we're doing. They can look at my photo gallery. One of my hobbies is photographing biologists. And so I have a photo gallery dedicated to biologists that are alive and well today, and they might enjoy that. And they can also find my email address there.
And of course, your pictures with hats.
Yes, and flowers too.
Wonderful. Well, listeners, definitely check out Spencer's website. And Spencer, thank you so much for joining us today and sharing a piece of your story.
Well, I appreciate having the opportunity to chat with you, and I've really enjoyed it.
Well, this was a pleasure for myself as well. And thank you, listeners, for joining us. We'll see you next time on another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.